This is TWIS, This Week in Science, episode number 590, recorded on Wednesday, October 26th, 2017. There was no patient zero. Hey everybody, I'm Dr. Kiki, and tonight on This Week in Science, we are going to fill your heads with lots of noise, parasites, and aliens. But first...
Disclaimer, disclaimer, disclaimer. The best evidence yet for aliens is that there are other planets, possibly inhabitable in a way we think that life may inhabit them. And that the number of these planets is not few, and that the conditions for habitability are not unique. Because in a universe of hundreds of billions of galaxies with 70 sextillion stars, there are so many possibilities that life on more than one planet is highly probable. And while our cosmic cousins are not likely, or are likely out there somewhere, it does not mean that we should claim that they have secretly fiddled with the formation of our civilization, just because some people can't comprehend how we got from knuckle-dragging apes to This Week in Science. Coming up next. Just terrible.
I've got the kind of mind I can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And good science to you, Justin, Blair, and everyone out there. If you're watching or listening to the show right now, it is TWIS-o-ween, at least where we are. We might be a few days early, but 'tis the season, right? And twist the season for spooky.
That too.
That's right. Twist the season of Halloween. TWIS-o-ween, exactly. I brought all sorts of science news. I hope you all brought it too. I've got promising prosthetics, patient zero truth, and aliens. What do you have for us, Justin? Yeah.
I've got creepy crawlies, parasitic plants, headless dinosaurs, and why bug eating isn't gross.
Not gross.
Not so gross.
Every Halloween haunted house I've ever been to maybe doesn't consider. I mean, I don't know. Blair, what's in the animal corner?
Oh, I brought two different animals driven to the edge of madness and
just like
devils. Devils outsmarting death.
Wow, the intro to the show is so creepy. I hope people can make it through the intro and keep listening because I think it's going to be a great show. Let's jump right in. We've got to start with aliens because is it aliens? It has to be aliens.
It's
right?
usually aliens.
It's usually aliens. Well,
that's the consensus. I believe.
Yeah, well, it's not
aliens find a way.
Not necessarily the consensus. So there is an article that has been published in the archive.org entitled Discovery of Peculiar, let's see if I can read this, Discovery of Peculiar Periodic Spectral Modulations in a Small Fraction of Solar-Type Stars by an E.F. Bora and E. Trottier from Laval University in Canada. They used data from the Sloan Digital Sky Survey to analyze the spectra of 2.5 million stars. And of those stars, they found that 234 of those stars have a periodicity, a signal in their light frequency. And it just so happens that that spectral frequency matches up very nicely with these F2 to K1 type stars, which are very similar to the sun. Dun, dun, dun. So, of course, they think that they've got an ET signal.
Yeah.
I mean, it's aliens. You got stars.
It is an extraterrestrial signal.
It's an extra, but it's not, they're saying extraterrestrial. They're saying evidence of extraterrestrial intelligence. And the reason that they say this is that one of the authors of this particular article, Bora, published in 2012 an article predicting a particular signal for extraterrestrial intelligence that would be caused possibly by other intelligences in space looking to meet other intelligences in space. You know, it's a lot like Tinder. Put out a laser signal, swipe right, swipe left, you've got a signal. Anyway, the light fluctuation fits into this kind of predicted range for a pulsation of a laser. Pulses of light separated by constant time intervals. And basically, the question is, you know, they set out to find, they predicted this. Like I said, if aliens are shining lasers into space to find other aliens, then we're going to have to look for this kind of a signal. Let's go into the Sloan Digital Sky Survey, look at stars, and see if we can find it. And they found it. So it was kind of like they found what they were looking for in the data. But that is to say also, you know, they looked at 2.5 million stars. So maybe this is a real signal. However, it's very interesting that that SETI and the breakthrough effort that's going to be listening, the Breakthrough Listen at UC Berkeley, they're going to be listening in space for evidence of aliens. They said, well, you know, all right, this spectral signal might be worth something. And let me quote from the response that was written on October 11th from Breakthrough Listen. They say there are the 1 in 10,000 objects with unusual spectra seen by Bora and Trottier are certainly worthy of additional study. However, extraordinary claims require extraordinary evidence. It is too early to unequivocally attribute these purported signals to the activities of extraterrestrial civilizations. Internationally agreed upon protocols for searches for evidence of advanced life beyond Earth, SETI, require candidates to be confirmed by independent groups using their own telescopes, and for all natural explanations to be exhausted before invoking extraterrestrial agents as an explanation. Careful work must be undertaken to determine false positive rates, to rule out natural and instrumental explanations, and most importantly, to confirm detections using 2 or more independent telescopes. So basically, okay, they say on a scale of like 1 to 10, this result is right now basically a 0 or a 1 on the significant scale of
1 is very significant. 0 is much, much less significant.
Yeah.
That's pretty wide scale there still.
Yeah, so the people who are busily looking for aliens have looked at this study and said, that's nice. That's a nice little study you put out there. Okay, we might look at those stars. We'll add a few of them to our survey, but basically need more telescopes. Sorry, you can't just say, but it's aliens. You can't just say.
Right.
But
However, this is what this show that we do is one part is sort of just the weekly sciencey news stuff, right? There's another thing that we do that I feel like is going to be archived well for history, which is if you could go back 100 years and 150, 200 years and listen to people talking about cutting edge science, you'd also learn a lot about what their view of the world, what they think is possible, what the perception is of the future of science. Like it would be as though we were listening in on science from, you know, couple hundred years ago, and people were like, Mom McGillicuddy's come up with a new salve. Cures more than just rickets. And someday there'll be a salve you can put on your chest for everything. And, okay, that's where they thought it was going. More salves. Like, there would just be more.
But we have that. It's called Vicks Vapor Rub, right?
Yeah, it's pretty great stuff, right?
Yeah.
But there's an element of whenever we do hear one of these stories that we're going to be archived back, by future people who are having trade deals going through with the alien extraterrestrials. Who are like, back then they just thought it was tomfoolery, and they're going to talk like olden time people too. That's going to come full circle. Mom McGillicuddy wouldn't be able to get her intergalactic salves if it wasn't for alien connection. But those yahoos in the olden times didn't believe it was really that possible. This first contact, if it ever is possible, or not, it wouldn't be necessarily contact, but reception of a signal from an intelligent alien species, is going to be just like this. Extremely dubious, right? It's not something that we're going to just, some people will go, aha, it's aliens. They have a whole lot of stuff. But the scientific community is constantly going to be like, you really got to prove that one. That one's going to need some seriously strong proof signal. I mean, we want video.
Everything needs extraordinary claims, require extraordinary evidence. And you can't just run out and say, I looked at some stars and some of them spin differently than others. And so aliens. You can't just say that. There has to be, you have to really rigorously prove or disprove all the other alternatives before everyone will start to get on board with you. And that's the thing, is that they went through archive and they are very sure of what they've come up with. They've said we looked at this, and so we can count them out. But other people have gone in and said, I don't think so. And Seth Shostak, in an email to another science writer over at astronomy.com, went so far as to say a lot of people didn't even think this paper should get published at all. So that's why they went to the archive. I mean, it's not good. It's not complete science. And so now people are coming out. It's gotten media because this is a media attentioning story. It's like, oh, aliens are out there, right? Everybody wants that. But no, science.
Science first. However, they could be right.
They could be. We don't know. And moving on. Moving on.
Scratching the surface of it now.
We are.
That's how it's
surface.
It's not going to be a giant saucer-shaped ship hovering over Washington that we go, oh, okay, now we have the first evidence.
Romulans.
It's going to be something really subtle. It's going to be something with a lot of dubious signal and noise.
Like
it's going
dolphins.
to be something like this.
Like dolphins. Exactly. No, I'm kidding. So
I thought that was a transition. I was like, oh, Kiki brought a dolphin story.
I didn't. I brought a story about the dubious nature of communication and media. Do you guys know Patient Zero? Not personally, but have you heard the story of Patient Zero?
Patient Zero is supposedly somebody who was, what's the word? When you have a lot of partners?
Promiscuous.
But took promiscuous to a magnitude of like, I'm going to set the record for being the most promiscuous person on the planet. And supposedly this person was like basically the first person with HIV, or the one that was the epicenter of the outbreak epidemic in the Americas.
Yes. But that whole story's wrong. The whole story's wrong. This guy, he was never even called Patient Zero by any of the researchers who were working on his case. And it's a very interesting story. NPR's run the story, and there are others. You can find the whole story in Nature, where it has been published by a number of authors working on it. 1970s, and Patient Zero, HIV-1 genomes illuminate early HIV/AIDS history in North America. In this Nature article, the researchers have gone back and looked at the early genomes, the HIV-1 genomes, the strains from whence North American HIV epidemic came. And they determined that No, there was never a patient zero. There were a number of people in the United States at about the same time who were infected, had multiple partners, and the spread of the disease occurred as a result of the
So this whole thing is an urban legend?
at the time. Yes. So let me tell you where it comes from. There was a book written by a journalist, Randy Schultz, who used a study that took place in LA. It was called the LA Cluster Study, where they were researchers were trying to get people in the homosexual community around that time to talk about their partners, name their partners, and basically form and create the network of where the disease had started, and to try and figure out how the disease was spreading and why it was spreading. He wrote a book called The Band Played On, and it's a best-selling book, or it was, on the AIDS crisis. And he got fascinated by this patient called Patient Zero. And Schultz tracked him down and basically made him out to be something of a villain in the book. You know, this guy who was going around infecting other guys.
Right, because that was the thing. It was supposedly the patient zero knew that they were infected and was willfully ignoring their own vectorness.
Yeah. So
it's always like made that patient zero sound like the most horrible person on the planet ever.
Yeah. And so what has come out is actually the CDC was investigating in Los Angeles, interviewing these men for the names of their contacts to be able to find the historical links between them. One of the men was called Case 057. He was an airline employee. He was a steward on airlines. And so he was flying across the country, and he was in every city he went to, having sexual contacts. And some of his partners developed symptoms of AIDS after he did, which would be expected. The CDC used a coding system because of their sequence, Case 057, because he was all over the place, and he kept ending up, because he was very promiscuous, he kept ending up in different, like, he couldn't be categorized as an LA identity. He couldn't be categorized as a New York identity. He became known as outside of California within the CDC database, and his nickname was abbreviated as the letter O. Case O.
Patient O.
Patient
Case
O. Yes, and it got misread by the media as Patient Zero, and the media ran with it, and the Band Played On story pushed it even further. So this is this Case 057, Patient O, Patient Zero. He actually helped very much. He remembered a lot of the names of his partners. His name was actually very memorable, so a lot of his partners remembered his name, whereas a lot of people who were having sexual contacts at that time didn't remember many of their partners. According to this article in Phys.org, over 65% of men in the cluster reported more than 1,000 partners in their lifetime. Over 75% had more than 50 partners in the last year, but they couldn't remember most of the names of their partners. Case 057 managed to provide 72 names of the almost 750 partners he'd had in the previous 3 years. So a researcher,
that's a really good memory. I don't. Like if I was trying to do this with customers who I've helped in the last year, I don't think I could come up with that many names.
Yeah, and so Patient Zero, in effect, became this known for infectious, became for the beginning of infection, and the first case, primary case, and index case just didn't work with the media, and so the CDC accidentally coined it, and it turned into a story. And it actually turns out that this guy was actually very helpful in trying to help the researchers. He was not going around trying to infect people. It just was a period of time in which people were unaware that they were passing diseases on, moving along in time, and it was not a lot of, it wasn't with intent. So he was maligned after his death in Schultz's book. And hopefully now that his story has come out, he will gain a little bit of his, gain a little bit of honor back. Mm-hmm. But there was never any patient zero.
Although we never knew his name, right? His name wasn't in the book.
It is in the book.
Oh, it is in the book.
His name was Gaetan Dugas, a French-Canadian man. And he was...
That seems in poor taste to use his name.
Yeah. Yeah. So now, it was a time and it was a place, and time has moved forward, and so has our understanding. And I think this is a lesson that we can all learn from. Something like patient zero, it strikes fear, and it strikes, we talk about patient zero for Ebola last year in the United States. We can talk about patient zero for Zika in the United States. We can talk about patient zero for a measles outbreak. This is a term that is now very, very in use, and it strikes fear into the hearts of people. And it's very unfortunate that it never really even existed to begin with.
Yeah, all because an O and a 0 look very similar.
Right?
They do, don't they? I never noticed that before.
It's a problem. I will often put the diagonal line through my zeros.
Right? Always put a diagonal line. It's not an O. It's a 0. Diagonal line. 0. Not. Not. N-A-U-G-H-T. This is This Week in Science. Hey, Justin, what you got?
In a cavern, in a canyon, in Sequoia National Park, was found a millipede with 400 legs, 200 poison gland silk-producing hairs, and 4 penises.
What?
Found in previously unexplored dark marble caves in Sequoia National Park, the bizarre millipede was sent to diplopodologists, which diplopodologists apparently are scientists millipedes.
Yes.
Bill Sher Paul Merrick, who immediately recognized its significance as evolutionary cousin to one of the leggiest animals on the planet, which is apparently found at about 150 miles from that site. The new species possesses only 414 legs in total.
Only that many?
Compared to its relative, who's the leggiest, at 750. Yet, it has a similar complement of anatomical features, if not legs, including 200 poison glands, multitude of silk-secreting hairs, and 4 penises. The new millipede, Lacme tobini, after cave biologist Ben Tobin of the National Park Service, described by its discoverer, Gene Kajaric. So this is... In addition to the new millipede's legginess, there's also bizarre-looking mouth parts that they say is of a mysterious function. 4 legs that have been modified into penises, a body covered in long silk-secreting hairs, and paired nozzles on each of its 100 segments that squirt a defense chemical of an yet unknown nature. This is truly a uniquely evolved creature at this point. In conclusion, the authors note that by exploring our world, documenting the biodiversity of this planet, we can prevent anonymous extinction. Something like this could have easily disappeared. They've only found it in one cave in Sequoia National Park.
That's amazing.
Do we know how big it is?
It's apparently pretty tiny. They describe it as thread-like, so it's apparently very, very small. I don't have the measurements here for it, though.
Interesting.
What's so fascinating is finding a creature like this. This is the new generation of exploring, where you can find a cave and find only one instance of of an animal that only lives in this cave and it doesn't live anywhere else. And we know that because of what we're doing, we'll probably kill it eventually, but we found it. So it's like a race against time.
Mm-hmm. Absolutely.
Yeah, if Sequoia National Park wasn't a national park, this thing would have been long gone.
Yeah, and so it goes to say, I mean, Blair in the Animal Corner has talked so much about the opening of new aquatic conservation ranges, of new national parks. We've talked about all sorts of the idea of not just conserving species, but conserving ecosystems. And yeah, it's this kind of a little animal that's a little frightening. But still,
it's not really so much frightening. It's
the
fascinating.
poison that comes out of that poison gland could turn out to be an ingredient in future medicine. There's the silk could be something that we use in manufacturing new technologies.
The other 200 nozzles that are putting out goo that they don't know what it does.
Yeah. See,
and someday we may learn that why it is a creature needs 4 penises.
I mean,
4 penises.
One can only
nozzles
imagine
with goo.
It's
all
dark
of it.
in there.
What are you doing in
Maybe.
there?
Yeah. And it looks like they're about 30 millimeters in length and about 1 millimeter wide.
Oh, wow. It's bigger than I thought.
There you go.
There you go. Wow.
Still very tiny, but
Yeah, 30 millimeters. Not very big.
not very big, but still much. That's pretty
200
long.
poison glands. Still not something I want to come across. Millipedes, not my favorite insects. Gotta say.
Not too worried about the poison from the millipede. I don't know why I'm thinking that's not going to affect me.
I prefer millipedes to centipedes.
Truth.
Truth.
Moving on, parasitic weeds.
Great.
Maybe able to steal genes. What?
Wait, parasitic weeds? Like a parasite, like a plant
Parasitic weeds.
weed, like a plant.
A weed that's parasitic. Well, we have parasitic. It's like an invasive species.
Oh, okay.
Right. But this one, they're discovering, there's a group of them really, are able to steal genes from the plants they are attacking and then use those genes against the host plant. So we've seen this before. This is sort of I don't know if it's a direct follow-up, but we covered something like this, about 4 or 5 years ago, where there's been evidence of horizontal gene transfer in plants. And this is specifically in weeds. And so this is something that we're used to bacteria doing this. Like, I have this gene. Oh, I could use a gene for that. Oh, here you go. And just hand it off, right? Kind of strange that it would happen in a more complicated system like a plant, but there are plants that can do this. They can take genes from a plant that's nearby and incorporate it in some way. However, this is the first time they've actually seen this gene transfer do more than maybe create a resistance to a bacteria or give it some little bit of a benefit. They're actually weaponizing, in a sense, these genes. So it says researchers detected 52 incidences of horizontal gene transfer from a host plant that later became functional into members of the parasitic plant family. And in this, they're studying the broom rapes. And this is researcher Claude de Pamphilus, professor of biology at Penn State. Although considered rare in plants, this is what they're seeing. These parasitic plants that we study from the broom rape family include some of the world's most devastating agricultural weeds. The horizontal gene transfer discovery is really part of our effort to try to better understand how parasitic plants work, how we can better control them. Our hope is we can use this information to find the best strategies to generate or breed resistant host plants. And so what they found, the researchers find, and they put this in, this is published in the Proceedings of the National Academy of Sciences, suggested this transfer could boost the parasitic plant's ability to invade the host. It can overcome defenses the host creates to try to ward off these attacks. And the horizontal gene transfer may also reduce the risk of infection for the parasitic plants. So the researchers are suggesting that the close feeding connections of the parasitic plant with their host may increase the chances of intact genes traveling from the host to the parasite's genome. And this is not just that they're planted nearby. the roots of the parasitic plant contact and even enter the other plant. And they begin extracting waters, sugars, mineral nutrients, and even nucleic acids, including the DNA and RNA. So it's not just that they're in the neighborhood.
That's what I was wondering was how are they doing
physically invading?
Yeah,
physically invading the plant.
They're not just neighbors. They're like, can I come over and get some butter? No, I'm just going to let myself into your house and your refrigerator. I'm just going to take it. And then your butter is going to define me.
Yeah, no, and then you know what? I like the way you have your kitchen set up. I'm gonna do the same thing over at my place, right?
I'm gonna do the same thing over here. Yeah, that's a good idea. You know, in fact, I think you should knock down a few walls.
Yeah. So this is and it's become this makes sense why there's an invasiveness to this strategy. This parasitic plants taking out crops across the world in different areas. If they're learning from the crops that they're attacking and overcoming their defenses, of course, they're going to be successful. Let's see. One of the most damaging sources of agricultural loss, it says here, is witchweed in sub-Saharan Africa. Astria.
Kevin Unique in the chat room is calling them alien weeds.
It's aliens.
Instead of invasive species, alien weeds. That kind of works the same way. We just have to hope that when the aliens really invade, they don't want our DNA.
But maybe this is the invasion. Maybe this is it, Kiki. Maybe they sent their plants up ahead first.
Oh, it's like an invasion of the body snatchers.
That's good.
Well, no, it's like a terraforming. It's like, we'll put these plants out there.
Mm-hmm.
Well, those are plants, right? Invasion of the body snatchers.
Parasitically invasive. Mm-hmm. What's the body snatchers?
Yeah, they grew people on plants. Yeah.
It was cloning. Yeah, they went from plant cloning directly into human cloning.
Yeah, and then there's Scott Sigler's book, Infected, his trilogy, the Infected trilogy. Those were spores from outer space.
Spores from outer space. Not that. No. Anything but that.
It isn't so. Oh. Are you good on that story, Justin? Did we cover it?
Yeah.
I think that's a fascinating story. Wow. It's like needles to the plants.
I still want to know how it does it, how it incorporates. I get that the DNA is there, but I want there to be a bacterial symbiosis thing. That's the level that the transfer is taking place, and that's then imbuing that to the plant somehow. But I guess they might not need that.
Well, it's a question. Are the plants defending against it, or if they're not putting up immune defenses, why not? Why are the plants that are being invaded and stolen from, why are they allowing it to happen? Is there, like you said, is there something that they feel like it's symbiotic?
Are you blaming the plant victim right now, Kiki?
No.
but it could be, that it's sort of telling the other plant that nope, this is normal. This is just part of you right here. Yep.
Yeah, so we need to learn mechanisms. What's the signaling mechanism? What's happening? We got to know why. How's it happening? But what
first story was 4 years ago. This one's not. Maybe 4 years from now we'll have that answer.
Yeah,
maybe sooner.
4 years from now. That'd be awesome. You know what's even sooner than 4 years from now?
What is it?
Right now, and it's Blair's Safari Animal Corner.
Loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
What you got, Blair?
I have a couple animals driven to madness. The first one.
I just noticed you got something behind you there.
No, there's nothing behind me.
No, on the wall behind you.
No,
Wait,
it's everything's fine. It's
okay.
So I want to talk about ants. So ants, they succumb to peer pressure. So let me ask you: if your friends jumped off a bridge, would you jump off a bridge too?
Nope.
Well, if they made it. I might.
What if they ate poison? Would you eat poison too?
Is that poison alcohol?
Yeah.
No, it's just
no.
straight poison.
No.
Probably.
Okay, so you're different than a carpenter ant.
No.
So carpenter ants, were discovered in a recent study from the University of Buenos Aires in Argentina to show that social information delivered by other ants overrides individual assessment of food. So the way that works, carpenter ants, they rely on sophisticated communication channels and on individual decision making. And they do that to make sure that their foraging is effective and that they find good food sources. The way they do it, pretty interesting, they actually swap food via mouth-to-mouth exchange, and that's called trophallaxis. So it's imagine telling your friend, oh, you got to try this cake, and then spitting it into their mouth. So that's how ants communicate.
So mothers and babies maybe sometimes engage in trophallaxis.
Sure, and actually that's an interesting point. That might actually help animals figure out what is good to eat.
Yeah.
Very interesting. So the ants use trophallaxis to do this mouth-to-mouth exchange, and that allows group members to pick up odors and tastes, which they then go to link with specific foods. So this research study looked at a laboratory experiment and a field experiment. They did both to see if a toxic substance, which they knew was strong and harmful to the carpenter ants, if they could make that food acceptable to the carpenter ants, if they associated it with an odor that is also found in food that they exchange via trophallaxis. So they exposed ants to toxic food, which was sugar water mixed with boric acid.
Hey, that sounds not good at all.
Yeah. And they mixed that with the odor they had previously learned about through this mouth-to-mouth exchange. And they showed that ants can detect whether something is toxic and that they will reject it as a possible source of food unless the odor they experienced was also experienced in trophallaxis previously. So even though they can recognize this toxic food, so this happens to such an extent that ants go on to then collect toxic food after they have been exposed to this toxic food that was also associated with this smell. So they essentially would commit suicide via toxic food based on something that they experienced from a member of their colony. So
I gotta taste it to believe it. And
Yeah.
then if you wanted to believe it, I really believe it.
If you wanted to kill a bunch of ants, then
Yes.
you wouldn't want a fast-acting poison.
Right.
You would want something that they could, somebody was going to try it, have somebody else try it, and then they'd go and collect it now. Now they're bringing it back to the nest.
What would make more sense would be to have two stages of ant poison. The first stage, not poisonous.
Yes.
Second stage, smells, tastes the same, poisonous.
I know that people do use boric acid as an ant poison. People will shake it on the ground around the outside of their homes. They'll put it in dishes within their homes, on windowsills, etc. And so this kind of thing, you put out a little glass, a little lid full of sugar water or little sugar cubes, and just let the ants come. Let them get used to it, and then switch-o, change-o. And suddenly...
Yeah.
High ant problem.
Yeah. So the moral of the story is that the desire to avoid toxic food is overridden by social information. So, so this can be extrapolated to many things. We can think about this. Think about how many people have acted.
People.
Well, of course, but I'm going to go there.
Anthropomorphization.
Anthropomorphization is generally something that is super fun to talk about, but not scientific. So I will mention real quick one of our favorite topics, the mob mentality. It's the perfect example of this. The mob mentality. You know better. You know better, but because you're surrounded by all these people acting a certain way, you act that way as well. So just a reminder to think for yourself.
And we also talked about the alcohol study a couple of weeks ago, where people drink more because they think their friends are drinking more.
Yes, yeah.
And so it's kind of that you're keeping up with the Joneses. Like,
absolutely.
Oh, my friends are eating French fries? I'll eat French fries. Oh, my friends are eating salad? I'll eat salad.
Yeah. And then a second story related to accidental suicidal tendencies in animals is from the University of Bristol, and this is regarding mongooses. So mongooses, as part of the weasel family, excellent sense of smell. They are themselves a predator, but they are also prey. And so they use their sense of smell to avoid predators, specifically by being able to smell predator poop. This study looks at mongooses in South Africa, and they were specifically looking at, bear with me, sound. So they were looking at sound pollution from urban environments, and they exposed these mongooses in the wild to these sounds. They had road noise playback, and these sounds negatively affected the mongoose's ability to detect predator smells. Even after they smelled the predator poop, the additional noise led to less information gathering and less vigilance. They overall acted way more vulnerable. So Professor Andy Radford from the School of Biological Sciences said, quote, "We've known for a long time that noise from urbanization, traffic, and airports can detrimentally affect humans by causing stress, sleep deprivation, cardiac problems, and slower learning." Again with the anthropomorphization. What's becoming increasingly more clear is that a lot of other species, mammals, birds, fish, insects, and amphibians, are also impacted in all sorts of ways by anthropogenic or man-made noise. So this is especially interesting because it is affecting the way their brain processes stimuli from a completely different sense. That is crazy. That their brain can't process smells the same because of sound pollution.
Well, I'm gonna ask you this: have you ever been in a noisy environment, or been trying to work on something, focus on something that's important to you, when somebody comes in and starts making a loud noise, or the stereo's playing really loudly and like jarring music that doesn't make any sense to you?
Mm-hmm. Yeah, I guess I've just never tried to identify smells while that's happening. But that sounds like a great personal experiment, is to tell somebody to start cooking something in the other room during a loud party and see if you can recognize what's happening. Exactly. Is that popcorn? Is that cookies? What is that?
But for I'm gonna say, for mongooses, the mongoose smell is very important because they, not only do they use it to find prey, but they also use it to, like you said, check out other threats in their environment. And so
I could find this. It depends on what
smell to be really messed up for them is one thing, but which senses in other animals are important that get messed up? What is it?
Yeah, personally, I would have. That'd be really interesting. But it would have to be, for me, it would have to be something that had voices. It would have to have talking. Like, I can put up with a tremendous amount of noise, music, other sort of things, and concentrate on what I'm doing. But I've got like this jungle ape hearing that's constantly on the lookout for vocalization. And I can't turn it off. I can hear somebody having a conversation outside and down the street as they're walking along, and I'm listening to them while I'm doing whatever. I have pretty good at this. I can hear two conversations at once coming from different areas even. But that can get in the way then, because I can't not hear it at the same time. I can't tune out people talking. If it's music, it's easy. But if there's vocalization in them,
Yeah, so it makes sense, the vocalization or whatever. If you're listening to a lecture or watching a video and somebody next to you starts talking, you're probably going to be distracted by the person talking, and you're going to have a hard time focusing. You're going to either want to focus on one or the other, and that's just a single modality, though. That's just hearing. But this is like all of a sudden, you're trying to smell your cup of coffee, and somebody starts talking to you next to you, and you're like, what am I smelling or am I listening? What am I doing? And that's the confusion.
Yeah, and so previous studies have looked at movement patterns and foraging, and those have definitely been affected by noise pollution.
Yeah.
You can wonder whether that's related to them trying to avoid certain noises or whether they can't hear other things. I think that's usually been the conclusion that's been drawn, which is why this is kind of exciting, because this is looking at processing of other sensory information disrupted by noise pollution, which in some ways, Kiki, you're dead on. It's the brain is the brain. And if the brain is having trouble processing information based on confusing, constant, unwanted stimuli,
right,
it makes perfect sense that it would make it difficult to process anything. It's just something that we haven't really looked at. And for so many animals, those complex sensory cues are exactly how they survive. It's how they find each other to mate. It's how they stay alive. It's how they find food. So if you take away one or multiple, or you just numb all these different sensory inputs, it's going to affect fitness of animals in the wild.
Yep.
So something to think about. If we all get electric cars, that'll reduce engine noise.
No, we see this. We see this in aquatic animals too, or at least aquatic mammals, whales and the like.
Sharks too. Yep.
Who communicate quite a bit normally, shut down their own communications when there's like large boat sounds or whatever going through. When there's acoustic pollution in the ocean, it really does affect everything they do.
Yeah. We'll see where this research goes and what we find out more and more. I mean, how are we affecting animals in the wild? How are we affecting their behavior and their survival? And then additionally, we're creating our artificial habitats, cities where more and more people are going to be, are living, and they are a jungle, and they are a very loud, raucous place. And so how are we even affecting our own survival?
Yeah. Absolutely. And this also affects the distance from protected areas that we think about having people, because we think about just creating physical distance, but not a lot is factored in at this point with light pollution and noise pollution about distance for protected areas. So that definitely needs to go into the equation as well.
Absolutely. All right, everybody. This is This Week in Science. It's time for us to go to a little break. It's time for us to complete the equation, right? We have another half of the show coming with many more stories. I've got promising prostheses, and then there's some, I don't know what other people. Justin, what do you have? I don't know. There's also some devils.
What is on Blair's backdrop? There's something. Is that a giant ant crawling down to eat her?
It's either a giant ant crawling down to get her, or it's a spider. That's a spider. It's a giant spider.
Largest spider I've ever seen.
Safari Blair. We're going to go on a break and find out what happens to Blair while we're gone. We'll be back in a moment. This is This Week in Science.
You heard with more than intuition. The line of reason shows the way to go. No conclusion. The methods of hypothesis and patience are the only things.
Hey, everybody. You know what I want to let you know about? I would like to let you know that we are pre-selling calendars for 2017. Blair's going to don't get attacked by the spider while I talk about this, Blair. We're pre-selling our calendars for 2017. You can pre-order them now. Blair's almost done with all the art for the calendars. We're going to be putting them in for an order soon. But you can put in an order now if you go to TWIS.org. They're $20 plus shipping, and I will let everybody know when you can expect the calendars after we have actually gotten them ourselves. But calendars are coming in if you want to follow along with science and TWIS for the year of 2017. Additionally, at TWIS.org, there's information about where we're going to be next week. You know where we're going to be next week? Next Wednesday, we're going to be here for a very truncated show, but our main show is going to be Friday next week. Next Friday, November 4th, we are going to be live in Baltimore, Maryland at the Baltimore National Aquarium. And we're very excited to be joining the Maryland STEM Festival for the Maryland STEM Festival opening ceremonies. And there's going to be a lot of stuff going on from about 1 o'clock to 4 o'clock at the National Aquarium. Our show starts at 2 p.m. Eastern Time on November 4th at the National Aquarium. All you're going to need to do is get yourself a ticket to the aquarium, and you'll be able to join us. Also, I hear that across the street is another science museum that will be having more opening ceremony activities from about 4 to 8 p.m. So there's a lot of stuff going on in the waterfront area down in Baltimore, Maryland, and we will be there and are looking forward to it. And I hear there's going to be fun. I think there's going to be sciencey fun. I hope that you can join us. Please come out if you are in the Maryland, Washington, D.C. area. It would be wonderful to meet you in person. Additionally, we have merchandise. If you're interested in supporting TWIS, helping TWIS continue to do wonderful, fun things that we like to do, help us afford equipment that we need, repairs that we need to make, web hosting, all of the things that we need to keep this show running and to be bringing you a show week after week after week. There are many ways that you can do it. Again, if you go to TWIS.org, you will be able to first, you can go to our Zazzle store. It's in the main header bar. Click on the Zazzle store link, and that'll take you to our Zazzle store. Direct link is zazzle.com/thisweekinscience, where you can find all sorts of logo emblazoned items that you can wear and share with other TWIS-loving friends. A lot of fun stuff there in the Zazzle store. Also, if you are inclined not for merchandise, but to help us out with donations, you can either scroll on down to the Donate to TWIS button that's on the sidebar. Donate, that'll allow you to donate through PayPal or credit card. And then additionally, we are using Patreon as one of our main donation platforms. And by donating to Patreon, you will become part of this wonderful community of people using Patreon. And there are rewards for donating. If you donate at the $15 or more per episode level, you will get one of Blair's hand-drawn pictures that are featured in the TWIS 2017 calendar. All sorts of goodies for those of you who join in on Patreon. If you are unable to donate, if you're unable to help us out financially, you can also just help us out by sharing us with friends. Tell your friends to listen to us on iTunes. Tell your friends to watch us on YouTube. Tell your friends that you think my Squirtle classes are weird and that they should come check it out also. But anyway, tell your friends. That's one of the biggest ways that you can help us out. We thank you for your support. No matter how you're able to support us, we thank you for it. We really could not do this without you.
Stop and think. The only real power that I do believe is the dollars and cents all these authors receive. If miracle wonders were held in their looks, why waste precious time and try selling their books? Why sit
and wait for your publishing royalty. If one has real power, who needs real loyalty? If you could travel by thought to a mystical place, why go to book signings and vie for shelf space? Why would you wait for your agent to call you in Venice and try to convince you he's worth 12%? Why would you bother with talk shows and crazed publicists? Why deal with the people that repeatedly insist that you show them real proof that your powers exist? Hey, convince some real skeptics or cease and desist. Trust me, 6 months from now, you folks will not be missed.
And we are back with more This Week in Science.
Yes, we are. And Justin, what you got? I'm curious.
This is a new publication on the bird-like dinosaur Avimimus from the late Cretaceous. Suggests they were gregarious social animals. Explains Gregory Funston, PhD student at Veneer School, University of Alberta. Our discovery demonstrates that dinosaurs are more similar to modern animals than people appreciate. Although the players are different, this evidence shows that dinosaurs were social beings with gregarious behavior who lived and died together in groups. So this is
dinosaurs gregarious. That's fascinating.
Yeah. This is coming from a site in Mongolia. First encountered by paleontologists about 10 years ago. The site contained thousands of shards of destroyed bone, laying the telltale evidence of a previous discovery by fossil poachers. After conducting additional field work, scientists discovered a bone bed with an assemblage of Avimimus dinosaurs who were extremely rare prior to this discovery. Funston explains that it is common knowledge that modern birds form flocks. And these flocking dinosaurs are exhibiting that same sort of behavior. With an assemblage like this, you can really understand why the dinosaurs died. You can't really understand why the dinosaurs died together unless you see the field site. We can tell that they were living together around the time of death, but the mystery still remains as to why. The paleontologists do know that the discovery highlights a potential trend of increasing gregariousness in social behavior in dinosaurs. Well, it's not increasing now. They're all dead. But these groups of dinosaurs became social towards the end of the Cretaceous. What remains to be solved is whether this increasing trend is based on dinosaur behavior or if it's because of how the fossils were preserved. So, dinosaur behavior towards the end of the Cretaceous? Is it a trend in the way that dinosaurs were dying? Or is it happenstance of how old the fossil finds are, and that's affecting the nature of what it is we can find? So this could be something, based on what he's saying, this could be something that is not just a late Cretaceous thing that's taking place where dinosaurs are becoming social. This could be how dinosaurs are all the way back, right? Yeah. It's rare to find this in the Jurassic and Triassic, but it is very prominent in the Cretaceous period. But this is the first time they found a bone bed of bird-like dinosaurs with this. So it's and
bird
then
behaviors had to come from somewhere, right? Or I mean, yeah. So this the what we're seeing in the behavior of birds or even of herd animals, really, according to this, both these groups coming from somewhat different sources, but the ancestral herd-like behavior or social behavior appears to have existed just fine in the dinosaur age. They weren't a bunch of solitary creatures going around being, mean to each other or whatever. Or they were, and then they were all dying out, and there was no food, and so they started flocking together, which is
that's also possible. Right? That's also possible. Yeah. Funston says that perhaps more important than the scientific findings is shedding light on the increasing incidence of fossil poaching and how this is affecting our ability to delve back into the past and find this sort of information. For this reason, he and co-authors have published their findings in the open access scientific reports, part of Nature group of scientific journals, inspired by their mentor, who's been working in Mongolia since the 80s. And they bring up, and this is the mentor, Curies, Students have taken up not only scientific cause, but also the higher social justice to protect our shared heritage. So they're sort of being a little socially active, politically active, I suppose, in saying this needs to be brought attention to. And so in this, they also bring light to actor Nicolas Cage.
What?
Well, yeah. Who apparently in late 2015, he won a Tarbosaurus skull. He actually outbid another star, Leonardo DiCaprio. They were both bidding on this Tarbosaurus skull. He won.
Then in the end, it belonged in a museum, right?
Well, yeah, he found out that it had been poached, that it was illegally acquired, and Nicolas Cage returned it. Yeah,
That's
he
nice.
returned
Good
it to
job,
Mongolia.
Nicholas. Yeah.
Yeah.
What did you do?
Way to go. Yeah.
So we should all, if we win a fossil, anything at any point,
yeah,
say, we should ask
one named
ourselves,
after him.
WWN do. What would Nicholas do?
Or name it after Nicolas Cage if it's a new discovery.
Yeah, so you can ask that about skulls, but don't ask it about like sound investments or anything like that.
Right.
Yeah.
There's that. I think this is it's a wonderful finding. Yeah. There's so much we still don't know. And it's just, when did these behaviors first pop up? Like you brought up the story last week of the cracking of the stones by the capuchin monkeys. When did humans first start doing these behaviors that we're so well known for? Birds are so well known for flocking. Fish are well known for schooling. When did these behaviors really get started and why?
Yeah. And I still like the idea very much that humans waited around these capuchin monkeys to crack a stone and then come up after. It's like, oh, we got some more. Okay, now let's wait for some more. See if they do it again.
It's very possible. I love it.
So it was for 250 years.
250 million years. I have a couple of cool stories related to the brain and the visual areas of the brain and plasticity. So the visual area of the brain has traditionally been understood as only... Whoa! What was that? Did a stick just fly out of the air?
I'm fine.
I think Blair just got attacked by a spider.
All right.
I think
okay.
I think Blair just got attacked. Sorry. Yep, there's a spider. The spider jumped.
I killed it.
You killed the spider. Okay, well, good for you, Safari Blair.
I mean, don't kill spiders, everyone.
Don't kill spiders. Spiders are good.
Spiders are nice.
Well, when they're like 2-3 feet across, maybe consider not hanging out next to them. Anyhow, the visual cortex, the visual system of humans, is known to not be incredibly plastic. We get our vision, and then at a certain point in our life, it becomes hardened, and the visual pathways are nailed in, and we see what we see. And if damage is done, there's no fixing it. Right? If damage is done, there's not a lot that's going to happen to fix it. Now, 2 studies out this week kind of really make us wonder whether this system is actually more plastic than previously thought. So one study reported today in Nature demonstrated that not you, but researchers can take embryonic neuronal cells and transplant them. So these neurons from the embryonic cortex transplanted into a damaged visual cortex of adult mice. They followed the trajectory of these embryonic cells and found that they actually integrated with these supposedly not plastic visual pathways, integrated and almost started fixing some of the damage. So they found that the cells actually started responding. These transplanted neurons reacted to visual stimuli that the animals were exposed to. And 9 weeks after transplantation, these cells were consistently responding more strongly to certain orientations of lines than to others. So this is just like developed neurons do in this area of the visual cortex. And it suggests that over time, over these just 9 weeks, the neurons' functions were finely tuned. which is pretty amazing. And so this question is now, I mean, the reason they did this is like, okay, can we take embryonic stem cells, can we take neurons and put them into the brain to fix damage in certain areas of the brain, like the visual cortex? Could we potentially fix damage to the visual system that has rendered people blind for extended periods of their lives? And one of the problems with this is we don't really know what's going to happen to the neurons once they get transplanted. And so this is one of the first studies that has really looked at this in the visual system. Yes, it's still in mice, but it's a very interesting result. And the ultimate goal is to transplant neurons in people. And this group that did this research out of Ludwig Maximilians University and the Institute of Stem Cell Research at the Helmholtz Center in Munich, Germany, their end goal is to actually to take neurons and to reprogram them. So to take stem cells and reprogram them, basically fix errors in the cells and then transplant them into adults. So in effect, be able to take cells from a person who's lost their vision, fix any errors or make any upgrades, and then transplant them into the visual system.
Wow.
Yeah. So that's their goal. And now the second study that was published yesterday in PLOS Biology is also really interesting. And this one is more interesting, though, because it is in people, not in mice. But it goes along with this idea of the plasticity of the visual system. It's an Italian study where they actually put chips into the retina of people with retinal pigmentosis, which is a disease that it destroys the retina and people go blind over a period of time. Now, they took patients who were blind and had been blind for a number of years, for a significant amount of time, and they put these retinal prostheses into their eyes. So these chips were taking information of light from the cones and rods in the retina and transferring information about light to the optic nerve and the cortex of the brain. And so the question is, okay, is the brain even going? It hasn't gotten signals for a long time. The retina has been dead. So do these nerves even work anymore? Is the visual system in these people going to respond? And what they found is that it did. And that people over time, and the people who trained more and trained harder on learning particular patterns of light were able to discriminate certain light signals better than others. And so the brain picked it up. These retinal prostheses are working.
Yes. This is fantastic news.
Oh, it's so exciting. This is like step by step. We are going to return sight to people who have lost it. And it's just, I get goosebumps. I mean, these are very beginning studies. This is not, not all the way along the pathway for sure. And the people who are seeing, they're not, seeing in the sense of all of the information that you could get from a working eye, of course, because you're dealing with just a chip that's implanted. But being able to get any idea of light information could help somebody navigate, could help somebody be able to getting more information than they have is better than none.
Yeah, of course.
Yeah. Nice. Yeah. So I'm excited about these two studies, and the fact that they just came out like a day apart. I'm just like, whoa, how exciting! Very exciting. All right, now, Justin, I'm talking about sticking chips and nerves into people's brains and eyeballs. What's up with bug burgers?
Well, yeah, the idea of eating bugs has been creating a buzz lately.
I got it.
So both in the foodie international development circles, more sustainable alternatives to cattle and fish, because we're overfishing things, and cattle they make a big footprint. Mm-hmm. And I don't know, maybe we'll find out that this ends up being worse. We don't know whether or not what the carbon footprint of insects would be in mass production, but we are looking for other ways to create sources for people to eat, and protein is one of those important things. This is a report appearing in the ACS Journal of Agriculture and Food Chemistry, examines how the nutrients provided by grasshoppers, crickets, and other insects really measures up to good old hamburger, right? And while eating bugs might sound appealing, you were doing a bit of squirming there, Kiki.
Yeah. I've had my share of mealworm flour cookies in my day. I can,
Okay.
Yeah.
So you're not completely, and it's long been included in a lot of diets in many regions of the world. These regions now are home to more than 2 billion people, according to the UN Food and Agriculture Organization. So there's a good part of the planet that's already used to eating bugs that might not have too much of an issue with this. The report also notes that about 1,900 insect species are documented as a food source globally. Source of protein, that's well established, so no problem there. But if the world is going to switch from cows to bugs in any great number, the creatures will also need to be giving us more than just protein. Iron, particularly important nutrient that is often missing in meat diets, causing iron deficiency, anemia, which can lead to lower cognition, poor immunity, poor pregnancy outcomes, and other problems. In light of these concerns, Yesemi Latinudada and colleagues wanted to find out whether commonly eaten insects could contribute to a well-rounded meal. The researchers analyzed grasshoppers, crickets, mealworms, and buffalo worms for their mineral content, estimated how much of each nutrient would likely get absorbed if eaten, using a lab model of human digestion. And the research says bug burgers could indeed fill that dietary need. That's going to work. Yeah.
Blah.
And as you've got there, what is that? Is that crickets and grasshoppers? You got way out in front in the amount of iron.
Yeah, the jumbo mealworms, the ones that you see most often, and the ones that the flour's made out of that Kiki you were talking about are the ones with the lowest protein amount there.
Oh,
that's
iron
pretty
amount.
high.
That's iron
Oh,
amount.
that's iron.
Yeah,
The
plenty of
mealworms-
protein, but this is the additional nutrients.
Yeah, the mealworms are very high in protein, but it's interesting to see that the crickets are way ahead of the pack and way ahead of beef.
Wait, no, that's the thing, right? Crickets, they say,
Move
have
over.
higher levels of iron than other insects and minerals, including calcium, copper, and zinc from grasshoppers. Crickets and mealworms are more readily available for absorption than the same minerals from beef. The results, therefore, support the idea that eating bugs could potentially help meet the nutritional needs of the world's growing population and actually may be a better source of food than beef. Right?
No, I don't like beef either.
But I'm sort of, but here's what I'm picturing.
Give me my cricket lentil burger. Come on, you guys.
The cowboy or the grasshopper boys of the future out there on the range. What does that look like?
The grasshopper boy out on the range would be probably in a lab coat in a building.
In a very large building, yeah. That, or we could genetically start breeding larger and larger, because cattle, I don't think were these giant bovine things that we're used to seeing nowadays. They were bred into this.
Oh yeah,
I
the pigs,
wonder how
cattle, everyone bred into it.
they were all smaller. How big do you think you could breed a grasshopper to be?
Oh, probably a couple of feet, I'm sure.
Wouldn't that be really cool? Like now we've got like these.
Oh my goodness! I mean, maybe
Ringing
they're
a
only
bell,
limited.
and here they come hopping home.
I mean, maybe like fish or like plants, they're really only limited by how much nutrient intake they can have and
Like rolling thunder on the
how long they
range.
have to live.
The late 2000 cricket herds stampeding across the American plain. Like, I'm all for it.
Yeah, chocolate cricket shake.
I feel like I've been warned about these giant crickets before.
You think so?
Yes, I put a link in the chat room. I believe there was an educational film about it.
Oh no.
And there was a version of it where there was a man and two robots who watched the film and spoke about it while it was happening.
Oh, is this the Mystery Science 3000 thing?
Giant grasshoppers. That's right.
Apparently
You
it's
always
called
have
The
to
Beginning
watch.
of the End. Name of the
I
film.
think it's going to be a fantastic future. And you know what? Really, when we're talking about eating the poison with mob mentality, how much worse could it be than your average fast food hamburger? Really, it's probably going to be an improvement. In fact, with the current manufacturing process, there's a decent amount of your protein and iron uptake that's probably coming from bugs anyway. What is it like?
Very true.
Amount of spiders in chocolate bars or something like.
Okay, let's not talk about that.
Why? Now we can talk about it positively.
It's good for you. That raisin, that wasn't a raisin.
Yeah. How about the shoe rubber in certain sandwich chains' bread?
Oh yeah.
I was just talking to someone about that today.
Oh, I haven't heard this. Oh, fantastic.
Yeah.
Okay, let's keep moving. Let's not talk about things.
What you got, Kiki?
Yeah, I got more neuron stuff. More prosthetics.
Kiki, you're going to talk to us about brains?
Yeah, I'm going to talk to you about prosthetics because that's what's really cool right now. Very interesting study out of University of Washington is they've put embedded sensors on the surface of people's brains, and those sensors allowed them to basically feel information. And so there's this problem with people who are paralyzed or have amputations. They can't, if you have a prosthetic or if you're using a brain computer interface to move a robotic arm, you don't have any haptic feedback. You don't have any of a closed loop of control to actually know how hard you're touching something. And you can think of how important actually the sense of touch actually is by how you have to consider maybe playing the piano or playing any kind of instrument like the guitar. If you could not feel how hard you're touching the strings of a guitar or the keys of a piano, there would be no nuance to it. You'd probably just be like smashing away at things and not really knowing the difference.
Mm-hmm. Or it would be like when I had nerve damage in my hand and I just dropped items all the time because I couldn't tell how tight I was holding on to things.
Right. So or if you had, say, an aluminum can, would you crush it or would you drop it because you couldn't tell the difference? And so this group, they have taken these brain surface electrodes to actually record brain signals and also stimulate back to the brain for sensory feedback. And so they had these people wear a glove that had electrodes in it. And this is while they were in the hospital for open brain surgery to begin with. They had their glove on and they were asked to hold their hand at certain positions of fingers being either close together or a certain distance apart without actually looking at their hand, but just by the sense of feeling how far apart their fingers were. And they would be stimulated by the stimulatory electrodes when they kind of got close to where their hand should be. And so they learned to use this sensory input that's actually like, wireless electrical information that's come from the sensors and actuators in the glove they were wearing that was actually getting electrically stimulating their brain to be able to help them figure this out. And people got very good at it. So this is one of the first times that we have a system where we have electrodes implanted into the surface of the brain, actually creating this closed loop bidirectional brain computer interface that could potentially allow a brain to control prosthetics or other external devices. And then additionally, a different group, they used a separate kind of concept in which, again, there was a different kind of electrode array being used to stimulate the brain, but on amputees, they put actual electrodes within the nerves of the arm that had been amputated above the point of amputation, so that any kind of prosthetic device that could be applied could potentially stimulate these electrodes and relay a signal to the brain of the person who's amputated. So in this study, what they were showing, this is from the University of Chicago and Case Western Reserve University, what they were essentially showing is how to stimulate the nerves. How many times did they have to stimulate the nerves in a row? How quickly did they have to follow those stimuli? How rapidly did the stimuli have to come to give the amputees any sense of pressure changing? You know, is something touching you hard? Is something touching you very softly? These people actually learned and responded to these signals very, very well. And it was a case study of 3 amputees in which they were actually able to figure out a lot about the process of how the nerves actually work to send information to the brain. And it could potentially be used with prosthetic devices and people who have limbs that have been amputated in the near future.
Mm.
Yeah.
Interesting. So So check this out. This is a story I had in the queue, but I wasn't going to do, but I'm going to do it now anyway. Did you know that apes are 50-50 right versus left-handed?
I think I heard that somewhere, but
yeah, I think we talked about it on the show a long time ago.
Yeah.
They're not predominantly left-handed or right-handed.
Mm-hmm.
And this is just all your handy talk got me thinking about this story again. 90% of humans, though, are right-handed. Mm-hmm. And in this study, they found a 1.8 million year old Homo habilis, which is the earliest evidence for right-handedness. And so this is, again, we don't have the numbers. We don't have a lot of these intact teeth that we can look at in a fossil of Homo habilis. They could be 50-50. They could be not so much. But so far, it's 100% right-handed.
And they can tell this by looking at the teeth?
At the teeth.
They're
Because
going
Homo
like
habilis,
this?
yeah, used their mouth as like another hand and would do things predominantly.
So these teeth would be worn down more if they're right-handed.
There's more labial striations, cut marks on the lip side of the interior teeth in the intact upper jaw fossil. So the earliest evidence thus far in the human lineage is right-handed.
Very cool.
It says handedness and language are controlled by different genetic systems, but there's a weak relationship between the two because both functions originate on the left side of the brain. Interesting. One specimen does not make it an incontrovertible case. There's more research is done. More discoveries are made. We predict that right-handedness and cortical reorganization and language capacity will shown to be important components in the origin of our genius and therefore the origin of the predominance of right-handedness in humans.
Yeah, I mean, again, it's if apes are 50-50, when did we become more dominantly right-handed than left? How long ago did it happen? Did it have to do with what aspect of the development of our brains? Say language? Did that lead to it? I mean, what is it? It's a question. It is a question that we will ponder until we find more information, until we find more fossils.
Yeah, we need more fossils now. Now we need some more fossils. And I can picture too the connection between language and handedness having to do with talking with your hands. Because I've never pictured like Homo habilis or Homo erectus or sort of gesturing while grunting. It's like right? No, but it's little showing like doing a hand gesture of a water buffalo running across the they could have done quite a bit of sign language as well as just vocalization, and that combination, if you started with a handedness or started with the side of the brain that was picking up language, could go a long way.
Yeah. All right, let's get to the quick stories at the very end of the show. I reported a little while ago on the FDA banning some homeopathic teething remedies. Well, it turns out the homeopaths are fighting back, and according to Ars Technica, have the NCH, National Center for Homeopathy, left a statement on its website blaming the media's exaggerated fear-mongering and, quote, dramatic headlines for getting the teething treatments pulled from the market. They are calling homeopaths to, quote, correct misconceptions, and said, despite these facts, that groups interested in seeing homeopathy destroyed continue to hammer away at the system, making exaggerated claims that create misunderstandings about and limit consumer access. And so basically they are trying to promote their own media on the other side to try and get the teething products back on the shelves, even though CVS has pulled all of its products from the shelves and Highlands has discontinued its products.
So what you're trying to say is
what are these products?
It's all rigged. It's a joke. It's garbage.
So when they're talking about teething products, the only homeopathic teething product I've seen that was at the newborn store here in town and was trending a few years back was amber necklaces.
Yeah, well, they're amber necklaces. That's different. What this is a homeopathic, which means that it should be a very dilute concentration of some compound, teething gel to calm pain and irritability. And the idea of homeopathy is, things should gain potency as they're diluted, but very often things are left in higher dilutions than they should be. There's no controls on since these are natural products, and so they're not controlled by the FDA really. So nobody's checking to see how much of these substances are actually in the products. And the FDA has said it found inconsistent amounts of belladonna in the teething products. Belladonna, which causes poisoning symptoms like seizures, vomiting, difficulty breathing, lethargy, excessive sleepiness, muscle weakness, skin flushing, constipation, difficulty urinating, blurred vision, and confusion led to more than 400 illnesses and 10 deaths that the FDA is investigating.
This is kind of sounding like those ants.
This is sounding like homicidal paths.
Yeah. So anyway.
Psychopaths. Don't give poison to babies. Just stop it.
Homeopathic treatments are not evaluated or approved by the FDA. That is true. This is a statement from Ars Technica, but this is true. They're not evaluated. Nobody checks to make sure how much of compounds of anything are in these products. And so even if they don't contain belladonna, they could contain something else. Even though these things are supposed to be dilute, they could contain things that are dangerous. And it's the question of what you want to trust your children. So anyway, this is the story reported on a week or so ago, a couple weeks ago, and now there's pushback. And then my second story is, remember the Mars Lander from the European Space Agency that we're trying to figure out what happened to it last week? Well, it did crash, and it's NASA's Mars Reconnaissance Orbiter thinks that it found it. It's a little black dot on the surface of Mars, and then a little white spot that might be its parachute. It crashed. But it's okay because it's not the most important part of the mission. The most important part of the mission was the gas monitoring orbiter that is working just fine, is in orbit, and will be looking at trace gases indicative of life activities coming from the surface of the planet. So that's something that is neat. But unfortunately, it is now starting to cause questions among those who were trying to push toward another more daring lander craft coming from the European Space Agency. So there are questions as to the future of landers on Mars coming from the ESA at this point in time. Rawr. And last story, Blair, you got devils?
Yes. So devils, actually the cutest of devils, the most vicious marsupial, the Tasmanian devil. We've talked a lot about facial tumor disease, and the latest update is actually quite inspiring. So recently I did a quick update indicating that Tasmanian devils, there's actually a small contingent of them that appears to have evolved resistance to this facial tumor disease. Now, a study from University of Tasmania, imagine that, monitored wild animals and actually found they looked at 52 devils. 34 of them contracted the disease, and 6 of those devils developed serum antibodies against the cancer cells.
Ooh.
Yes.
Awesome.
Yes. So they healed themselves from the facial tumor disease, which is huge, not only for the devils, because these animals might actually come out of this 80% die-off from facial tumor disease since the 90s. They might actually come back from that. But if we can find an animal that can cure itself, that can develop antibodies to cancer, that is definitely something we can use.
Mm-hmm.
Absolutely.
Mm-hmm. Yeah.
Unfortunately, had to take a majority of the population to fall prey to it before it.
Right, but I mean, that's
good,
how evolution works, right?
Yeah, talk about a population bottleneck, though.
Yes, absolutely.
But this is good. This is a good direction. There is hope for the devils.
Yes, there is hope for devils, which they currently have about 20,000 individuals. So that's not terrible. It's not like with elephant seals where it was less than 200, or it's not like cheetahs where there's less than 50, or it's not like the Mexican gray wolf where it was 7.
Or humans where it was 1,500, they think.
Yeah,
absolutely. So 20,000 individuals is something potentially we can work with.
Pretty good. Pretty healthy.
Absolutely.
Fingers crossed for the devils. Fingers crossed for some cancer enlightenment from all of this.
And in the meantime, this is going to bring an end to our TWIS-o-ween show. I hope you've been entertained by the devils, the bugs, the things in your brains, all the fun science that we brought for the show tonight. And until next time, I want to give shout outs. Thank you to our Patreon sponsors. Thank you to Chris Clark, Paul Disney, G. Burton Lattimore, Dave Friedel, John Ratnaswamy, Richard Onomus, Byron Lee, E.O., Jared Lysette, Ulysses Adkins, Kevin Parachan, Andy, Keith Corsell, Jake Jones, Mike McGraw, James Randall, Eric Schwab, Bob Calder, Mark Nazaros, Ed Dyer, Trainer 84, Brian Hedrick, Layla, Marshall Clark, Charlene Henry, Don Kamarechka, Larry Garcia, Randy Mazuka, Tony Steele, Steve DeBell, Haroon Sarang, Melissa Mosley, Alex Wilson, Jason Schneiderman, Rudy Garcia, Greg Guthman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts, Craig Landon, Daryl Lambert, Rodney, David Wiley, Robert Aston, Darwin Hannon, Cosmic Gypsy, Brian Hone, Orly Radio, Brian Condren, Pixelfly, Marked, Nathan Greco, Hexator, Deborah Smith, Mitch Neves, Flying Out, John Crocker, Richard Porter, Christopher Dreyer, Ben Rothig, Sylvan Westby, Artyom Shuwata, Dave Wilkinson, Steve Mashinsky, Rick Ramus, Garen Swinsburg, Phil Nadeau, Braxton Howard, Salgood Sam, Matt Sutter, Emma Grenier, Philip Shane, James Dobson, Kurt Larson, Stefan Insom, Michael George, Russell Jensen, Mountain Sloth, Jim Drapeau, Tara Payne, John Maloney, Jason Olds, James, Paul West, Alec Doty, Aluma Lama, Joe Wheeler, Dougal Campbell, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie, Paul Stanton, David Simmerly, Tyler Harrison, Columbo Ahmed. Thank you for all of your support on Patreon. And if you are interested in supporting us, you can find information at patreon.com/thisweekinscience. Also remember that you can help us out simply by telling your friends about TWIS. It's so easy. Share the science love. And on next week's show, remember we are going to be live on Friday at 2 p.m. Eastern Time at the Baltimore National Aquarium for the Maryland STEM Festival. We'll be live there in person. So if you're in Baltimore, you can come join us at the aquarium. If you are not in Baltimore, we will be live streaming. So we hope that you join us at the show any which way you can. And additionally, don't forget, you can pre-order our calendars for 2017 over at TWIS.org. Oh, yeah. And, there are the websites. You can go find us at twist.org/live, where you can watch live if you want to watch it live next Friday. Next Friday. November 4th. Next Friday. November 4th. Twist.org/live. Watch. Join the chat room.
What time?
2 p.m. Eastern time.
2 p.m. Eastern time. That's 5 p.m. Wait, no.
5 p.m.
It's
Pacific. Earlier here.
No,
it's the opposite.
11 a.m.
It's 11 a.m.
11 a.m. Pacific time. There we go.
11 a.m. Pacific time, Friday, 2 p.m. Eastern Pacific time, Eastern Atlantic time. What is it called again? Just Eastern time. 2 o'clock.
2 o'clock. 2 o'clock Eastern time. That's right.
Friday, new time. We won't be here. We won't be here at the regular time. So if you come back at the regular time next week.
We will. Just for we gotta talk. We gotta talk about what we're gonna do at the show.
Are we doing a pre-record?
No.
We could do. Okay, we'll talk.
We're doing a pre-meeting during the show time. So
very cool.
On the show. Yeah. And if you aren't confused enough right now, just make sure you catch our past episodes at twist.org/YouTube or just twist.org.
Yes. Thank you for enjoying the show. TWIS is also available as a podcast. Be sure to Google This Week in Science in your iTunes directory to find all the iTunes shows that have been archived over the years. Also, we're on the mobile devices. TWIS for Droid, that's TWIS, the number 4, Droid app in the Android marketplace. Or simply Google This Week in Science and anything Apple Marketplacey, and you'll find our stuff there.
For more information on anything that you may have heard here today, show notes will be available on our website. That's at www.twist.org, where you can also make comments and start conversations with the hosts as well as other listeners.
Yes, or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in the subject line of your email, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, but in Baltimore. And we hope you'll join us again for more great science news.
And if you've learned anything from today's show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just get understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And this week in science is coming your way. So everybody listen. listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science.
Blair, be careful. Blair, don't make any sudden movements.
This Week in Science. This Week in Science.
Don't make any sudden movements. Blair, be very careful. I believe there's a cobra on your pith hat, dear. I don't know if you shouldn't make any sudden sounds, but just you seem to be moving your head about quite a lot. Now, quickly, do you have a recorder? Can you play it a tune? Yes. Oh, yeah, get it to bite something else. Oh, no, you had it there for the moment.
Yeah, that snake on your hat, just whack it with a stick. Just do that. Just whack it with a stick, Blair.
There you
That'll
go.
make it happy.
There you go. Yeah, anger it.
Pikachu!
Bless you.
I think it's gone.
I really need to take off these Squirtle glasses. They're squeezing my head and my eyes are hurting because I've been in sunglasses indoors for hours. Good gracious. It's the after show. I can be less squirtly.
Yeah.
You guys, I think it's safe. I think it's safe now.
I can work with this wig. It's good.
I like it.
Good cut for me, right?
Is that like a Leeloo wig? What is that?
I know it's kind of the right color, right? It's oh, what's her name? Marcy. She's a Pokemon. She's one of Ash Ketchum's traveling partners.
Mm-hmm.
Yeah, so I have red suspenders and I guess a yellow tank top when I get to wear jeans, which is awesome. Any Halloween costume where I get to wear jeans. Perfect. I know, right? Multipass. Leeloo Dallas. Multipass. Oh, zombie.
So I'm now
density.
very excited to go to Baltimore because it turns out
I can do that.
I'm.
That's a good point.
It turns out I'm from there.
You're from Baltimore?
What?
I'm from Baltimore. I had no idea.
What do you mean? Is that where your family
So this is an unsolicited.
came across on that?
Well, yeah. Maybe I haven't found the actual boat that they got here, but I've got folks that were. I've got somebody who was John Baltimore Harriman back in 1671 to 1711.
Just because his name was
and a whole
Baltimore doesn't mean he was from Baltimore.
No, he was in Baltimore, as were like a whole bunch of the other family, the Simpkins, the Nortons, the Masons, the Webbs, the Short. They like there's this like I did the Ancestry.com free thing, like free trial dealing, and it's crazy. I've got a couple people who fought in the Revolutionary War already. I've got one family line that went back all the way to like the 1500s. It's traced back to France. late mid 1500s. Like this thing is crazy. It is so wild. And what's also very interesting is not only the size of the families. People had a lot of kids. It was not uncommon to have like 7 or 8 children. But also the fact that
Very
people
common and safe because people died.
There's a lot of, there's a bit of that going on there. But it's also the names. You notice that names, you might say like, well, I had a grandpa so-and-so. I think I'll use that name to name my child, put that in the name, maybe as a middle name or something like that. When you do that, what's crazy is that it makes your entire family line look very uncreative. Because chances are that name wasn't just their name. It was a name before and a name before. And there's tons of, first sons with the father's exact names and stuff like that. Name trends that go about. My Italian side is actually quite boring. If you go back, it goes, there's it's an Angelo, there's a Frank, then there's an Angelo, then there's a Frank. Like they kept the same names like over and over again. Of where you are from in the Western world, there's, I think, about a 40% chance that your name would be Mary if you were born before the year 1900. A lot of Marys in the world. My goodness. But yeah, now I'm going to get to go. There's apparently still I looked up. There's apparently a Harriman restaurant in Baltimore. That maybe some of my ancestors never left, and the Harrimans are still cooking. I don't know. It's gonna be interesting. I've got this list of names I'm gonna look for if we do any look at any historic sites. I'm ready now. I've got this list of that's my great something or other. Fun. But yeah, I have a whole bunch on one side in Baltimore, and then another line is like pre-revolution. I have a lot of colonials. I did not know that I had this many colonials. There's a ton of I'm very colonial past. There's not a whole lot of people coming off boats. Part of this. New Jersey is also a big colonial hotspot. And then for whatever reason, a lot of my family moved to Missouri and hid there for about 100 years. And then we came to California. During the Civil War. Sort of gold rush times. There's a lot of gold rush to the Californians.
A lot of gold rush to the Californias. I think that's why
Strengths
my family came out.
Strengths wants to know if you have any conflicts in your any convicts.
Convicts.
Not that I can tell. So if I now there could be plenty in extended family. Because while each of my relatives has a list of brothers and sisters, I'm not tracing those families down through every. I've got some a lieutenant something or another in the Revolutionary War. A bunch of people who are in the Sons of the Revolution or Sons of the Revolutionary. What is it? Sons of the American Revolution. There's pensions that get paid down to their wives as widows and their children, and in some cases it even looks like grandchildren are getting some sort of government benefit for their fathers and grandfathers having fought in the Revolutionary War. It's kind of interesting. I've got people on both sides of the Civil War, but most of them took off. Most of them kind of bailed. A lot of them kind of took off while that fighting was going on to go live in California, kind of get away from it there. What's really interesting is there was one ancestor that was private. And you can mark private, which you can kind of, so that, people can't see who that is in your family history. And normally they do that for people who are still living. Just so somebody else can't go and look up their family history, can't connect them to it, I guess. Because they're alive and, you don't want to, I don't know, give out somebody who's living's personal information. But to find one that was back, great. Where's this guy? This guy is in. Oh wait, no, I don't have his total info here. But this would have been somebody. Oh gosh, who would have been born probably in the early and mid 1700s. Document with this name. Now it's kind of sketchy because you get certain documents, but you have to put them in the right time and place and make sure that there's not somebody else in that area with the same name. And so there's little documents that you can actually bring up pictures of. And this was under wills and notary stuff. And so I was like, oh, well, a will would be perfect because you're bequeathing stuff to people. So you could really identify if who this person's children were based on or spouse was based on a will, and that could confirm their place in the family line. And only it wasn't a will; it was a record of sale of a young Negro girl. So, yeah, there's, and it kind of pissed me off that people were making that relative private. So that people couldn't see that, like the family shame of it or whatever.
Yeah,
but I'm like,
hide it.
you're then hiding not just your family history. You're trying to hide history at that point. You're trying to hide actual history. So yeah, there's that. There's interestingly, like back east, and this is back in the Baltimore region. There is a fair amount of photographs of headstones. So you have this person and you might have a birth record, you might have a voting record, you might have a census record or something like this. And then, they have about dates when they were born and when they died. And then you'll get a tombstone that'll be that person with the right years and the exact dates of their death and their birth and, survived by a spouse or married to or whatever, and another bit of information maybe even on the tombstone. It's a fun puzzle. I suggest everybody do the Ancestry.com, at least if not the DNA thing, which I might end up doing. The family tree is a really fun puzzle if your peeps have been in a country just a few generations, which is what I was trying to look back into. It was just a couple of generations. It ended up going crazy all the way, sort of disappeared. And then you're like, well, how did they get there? Where did they come from? And maybe that's like my Italians. I don't have like an international thing. So my Italians end when they arrive in the United States. That's as far as back as they go. And that's like the mid or late 1800s. Pretty much all the Italians that I've got here, just about late 1800s. And then it's just a few great grandparents that their family history unlocked all the way back. And so I had a fair amount of collection of documents, photographs, names, that sort of thing. I mean, I had photographs that have, thankfully, names of who's on them on the back, which helped me place names of great-grandparents, siblings, right? So I had a nice little collection. And then somewhere in everybody's family is this person who's hoarded the family information. They have that Bible with the list of names in it, right? They have a family tree that was compiled maybe a couple generations ago that they still have possession of. And so even if you don't have anybody in your immediate family, even your grandparents weren't that person, you may have a great grandparent who or a great, what would it be, like a third or a fourth cousin out there somewhere who does have a collection from a sibling of your great grandparent that was one of these collections, right? And so part of what you can do in this is compare other people's family trees. And what's really fascinating about that is, of course, they might not be interested at all in your grandmother, right? And identifying who she was in her life. And they might have the date of her birth, but not the date of her death, and they might have her name spelled wrong. But they've got all of the siblings, they've got the name of those parents, the parents of the previous generation, and they've got towns that they lived and other bits of information that correlate, but also fill in blanks where you've got blanks. So it's sort of this group effort of people connecting the information that they have of their family and finding other families that can fill in the information. So as a, for instance, and going back 4 or 5 generations, there's people I've never heard of. But I can get right up to it. And there's a bunch of other people who can get right up to it, right? Right close to the generation after, say. And because all of our information is overlapping of at least the name of a parent or where they were from, is all somebody else needs to do is know where that person was from. And then suddenly we're making these connections back a whole other generation. And that person might have had a better documented existence with census data, with military records, with this sort of thing. And so they're already flush out and easier to connect back to another generation. Having a tremendous amount of fun, losing a lot of sleep with this thing because there's a fair amount of it. I was looking, I'll tell you a funny example too, because the computer I think they've scanned in census documents for the every 10 years going back, over 100 years. They've scanned it in, and you can tell that the computers have some a little bit of deficiency in reading old handwritten cursive script. Trying to find there was this Colum Jackson, Colom. I kept trying to look for this Colum Jackson. It was supposedly my great grand something, right? And I kept looking, and then because it was another family tree, so I assumed I finally found the document that said that there was a Colum Jackson, and it turns out if you read it, it's Calvin. His name is Calvin,
not Colin.
And it was
Calvin. It's
not Colin, which I was like, and I even researched. I'm like, what is Colin? And like, it's a last name that's particular to the South. A lot of people have this last name, Colin. I'm like, oh, well, maybe this is a real thing. But then when I saw the actual document, you can actually pull up and see the census document from whatever 1840 or whatever it was. It's obviously Calvin. It's clearly Calvin. And so then, now that you have a right name to punch in there, Calvin, instead of Colum, might have other ties. He might have done business. He might have a gravesite. He might have a birth record. He might have his children have a birth record. And then there might be a spouse there, right? And so then suddenly you're building a branch of your tree that kind of was stuck because there's no such thing as Colum. Yeah, very fascinating, very fun.
Digging into that stuff is always fun. Like family trees.
Yeah.
Following the family. Let's see. Is there anything that we need to think about for next week? You guys have your travel stuff figured out? I mean, we can touch base on stuff like this Wednesday for sure.
Well, you saw our group text, so no.
Oh, you still haven't figured that out. Okay.
But I know that I'm getting on public transportation on that day to get to Oakland Airport. That I know.
Awesome. Get to the airport. That's it. Just get to the airport.
Yes. And if Justin wants to do that with me, he will tell me.
Yes, I think I will. That seems like a good plan.
Okay.
Awesome.
So we just have to pick a rendezvous time and place.
Yeah. You guys figure out your rendezvous.
We shall.
And hopefully I'll see you guys
in Baltimore. Do you land around the same time as us?
Yeah, I think I land an hour later. If you guys can hang out for an hour, we could all take a cab together because we're all going to the same place.
Oh, yeah, absolutely.
Yeah.
We'll grab a beer or something in the airport.
I'll give you my flight information so you'll know whether or not my flight's delayed or anything
Blair,
like that.
don't move. Blair, don't move.
Back.
Blair, do not move. On your right shoulder. Don't look.
You need a mongoose.
Don't look. It's your other shoulder. But don't look.
Totally need a mongoose.
I think your snake is back.
Ricky Tikki Tavi, where are you when I need you?
I don't have any mongoose in here.
Ricky Tikki Tavi, where are you? Let's see. We'll figure out stories.
Yes.
We don't really need that many.
Did you guys see my email? I found one.
Yes, I saw that you found one. That's awesome. It's a great story.
Pertinent to the aquarium.
Yes, which will be great. We have an aquarium interview I've been informed that we're going to have somebody from NASA for an interview, but I need to talk with Phil to see if that's going to be confirmed. If it's not, we'll either bring more stories, like one more story each or something, or we will just interview people from the audience at random. Or I'll find somebody for an interview. And then we have our musical guest, Shodake. Just so much fun. Shodake is going to be so much fun. We're going to do a demonstration. He's into science and music and fun stuff. Yeah. Vocal percussion.
Wait, beatboxing?
Yeah.
Oh,
Shodake
wow.
is a beatboxer.
Okay.
Our musical science and music is going to be beatboxing.
Very nice.
Yeah, it's going to be really good.
Just no freestyle rap contest, please.
No.
What? Why? I was all prepared for the thing.
Then you can do it
Ready
by
to
yourself.
freestyle?
You can rap the disclaimer, perhaps.
Oh no. Maybe you could. He could probably give you some beats. Let's say,
Mhm,
Shodake, give us some beats.
mhm.
From the Big Bang to the...
There we go. Yes, good. I like it.
Are you supposed to clap for rap? Is that how that works?
Just little
With
claps.
one hand. One
Your
hand.
fingers.
Sir, good rapping. I say. Top notch.
Thank you. Thank you very much.
I'm-
Thank you.
I'm
Thank
clapping
you.
like my anime character.
Thank you. Thank you.
That's right. That's right. Yeah, it's going to be fantastic. So we've got a place to stay. Thank you to Patrick for helping us out with that. It's going to be amazing. Yeah, I think we're going to have some fun. Our show's going to be done by like 4 p.m. And then we've got like the afternoon.
Well, and I don't know
Hang
if
out.
you're both aware, but we're going to the zoo the next day.
Oh, we are. That's very far away, though.
The Maryland Zoo?
Isn't it very far away?
I don't know. I didn't look into it yet. I was kidding. I'm going to look into it.
I'd go to the zoo. That would be super fun. But
Well, because they have
yeah.
the polar bear stuff for STEM Fest going on the next day.
the next day.
Mm.
That's right.
So, depending, if it's near something else that we also want to do, we could do that. So that reminds me, actually, I'm going to see if I can reach
Zoo.
out to people at that zoo. And say,
Oh, in Baltimore. Maybe it's not that far away. That's awesome.
Mm-hmm.
Oh, it's near Johns Hopkins. We can do the zoo and a little trip to Johns Hopkins. I know somebody who works in the nano science department there, the media.
Great.
But that is, let's see, I see where that is. It's ish, 20, 30 minutes probably from Inner Harbor.
I don't know what these words mean.
You wouldn't want to walk there.
Okay. Maybe you don't want to walk there. Maybe I want to walk there.
No, you don't want to walk there.
Okay. Okay.
Yeah, I did some walking in Baltimore once upon a time. And then I found out that I shouldn't have. People are like, oh, where were you walking? I was like, yeah. I apparently walked through a very dangerous neighborhood on my way to get crab. Just wanted crab for dinner, man. Let's see. Yeah, so we'll figure out what we're gonna do. But I think we have a little meetup maybe on Friday evening.
Yeah, that's going to be great.
Little science meetup Friday evening. And anyone out there in Baltimore doesn't know about the meetup, message me on Facebook and I'll see if I can get you information. Oh gosh, Blair, I just ran across your pictures for tonight.
Yeah.
I love it. Oh my God. Gotta get better at the pictures thing. Somebody just sent me this story that's awesome. And I don't understand it necessarily because I haven't had a chance to read it all the way through yet. But published in PLOS One, some researchers created a dough out of 237 cubic centimeters of water, 355 cubic centimeters of flour, 133 cubic centimeters of lemon juice, 59 cubic centimeters of table salt, 15 cubic centimeters of vegetable oil, and 2 cubic centimeters of food coloring. And you could probably put sugar in it and make it a crust. Anyway, according to this article, they formed it into round blobby shapes and then hooked them to a jumper cable and measuring devices with a red LED positioned close to the blob of dough. They exposed the dough to electric shocks paired with flashes of the LED. And this is similar to how they train nerves like in aplysia for conditioned responses. In some trials, the shock and the LED flash were simultaneous, and in others, the flash was delayed by up to 500 milliseconds. Then after training and following a 1 or 5 minute delay, they exposed the dough to a flash of the LED and then observed the dough's spectral power density. So basically, the conditioned dough, when it wasn't electrically shocked again and was only presented with the LED light, it produced the same amount of electrical activity as it would if it were just shocked. Untrained dough did not show the same response.
What?
According to this article in PLOS One, they trained a blob of dough.
I don't like it.
No, I know. Conditioning in a blob of dough. And seriously, I gave the recipe. It's a blob of dough, electrodes, and a red LED light.
Yeah, is this for real?
It's okay. Yeah, I'm screen sharing right now. I'm going to take you to PLOS One. Let's see if we can find the actual. Okay, so that's just the PLOS One link. So here's the study, a new study. That's the one I should have clicked on. A new study. Experimental evidence of classical conditioning and microscopic engrams in an electroconductive material. And what makes it electroconductive is the fact that it has lemon juice in it and salt.
Mm-hmm.
Lemon juice and salt. Anyway, yeah, they conditioned the dough, and the dough either responded or it did not respond. And let's see if we have some other. And then they mushed. And then another part of the experiment, they mushed the dough all up. So they had a control, they had conditioned, they mushed up, and then just light and shock. And basically, yeah, if it was conditioned, it all responded the same. It's very. So what they say, this isn't learning, complex learning per se, but if you have a material that's structural integrity can be changed by some sort of input, then hypothetically learning is a much more basic concept that can be shared much more widely across organisms. Yeah, and what they say, let's go back to that article. They say in the article, that learning, in summary, the data indicate that a conditioned response can be encoded into a simple material, that the conditioned response is associated with structural modifications within the substrate. So maybe learning is way more fundamental than anyone ever thought. But seriously, a ball of dough responding to light.
Did you just say learning is fundamental?
More fundamental. Very fundamental. Learning is fundamental. But anyway, yeah, this does make my brain hurt. I don't really.
Yeah.
I'm like it. Yes, the data you did, and I don't understand. How on earth did the light affect the dough? That's what I'm wondering. I understand the
Heat?
electrical. It's an LED. They put off minimal heat. So that's, I mean, it was a red LED too. So it was like a red.
So aliens is what you're saying.
Yeah. Yeah.
Mhm.
Aliens. Yeah. Totally. Totes aliens.
Spores from space. Got it.
Spores from space.
Yep.
Spores from space.
Yeah. Weird. Very weird.
Ha, getting a homeopathic vibe here. Right, Salty has. It's either homeopathy or it's that being mean to water thing.
Being mean to water.
Don't tell me you haven't heard about these experiments.
I have not.
All right. Yeah. Mean to water.
You what? You what?
Yeah. Hold on. Power of thoughts. Emotos? Is it Emotos? Ha ha ha. Good luck on that email, Fada. Have a good night. Yeah, let me see. I think Masaru Emotos. I think it's Masaru. Water crystals. That's what it is. Water crystals. Okay. Let's see if I can make sure I find a good. Just use his website. So let's see if he has pictures. Basically, he played different music or was mean to water and then froze it into crystals and took pictures of the crystals that were formed. And this guy, Masaru Emoto, and he showed that if you played nicer music, the crystals were prettier. So a heavy metal
get out
song.
of town with this. Get out of town with this.
Yeah, a Korean folk song was kind of weird. Amazing Grace was beautiful. And look at Edelweiss. And then, I mean, you get like Farewell Song from Chopin. And what is that? Heartbreak Hotel from Elvis. What? But then John Lennon's Imagine. Wow.
So was this replicated ever?
Oh, yeah. People all over the world have been... yeah, and so another one where they showed the water photos. He showed the water photos and made it into crystals. Like he showed the water a photo of an elephant, and the water loved it. And then there were
God,
prayers that they did to the water. So Chi of Love sent remotely by 500 people.
They prayed to the water, and this affected the crystals.
Yeah, and then they had different words that they gave to the water. So eternal or evil, love and gratitude.
Hemp got a nice, beautiful one, didn't it? Yeah.
Is that in there? Yes.
Yeah.
Yes.
So it what's really fascinating about this, actually.
Did the researcher get some hemp as well?
Well, what I find very fascinating about this is that the crystals actually seem to understand English.
Mm-hmm.
Exactly.
And have an opinion.
Exactly.
Based on that,
Mm-hmm.
that they then communicate and articulate through different
I would guess that
Right.
the people saying it know the context of it and therefore carry spiritual value with the words that they speak to the water.
Yes, and so it's resonating or some kind of vibration.
So for the word evil to look like the evil crystal, wouldn't you have to have somebody with evil vibrations in the room at the time? I mean, you couldn't just like when I say evil, I'm still like having my positive vibrations in the room. I'm not bringing evil into the room, so why would that be different? But when I'm thinking of hemp, it's really this beautiful, intense crystal. It's kind of automatic.
Yeah, and he made a documentary, and he did a rice
Oh my God.
experiment, and like all
Can
sorts
we
of things.
please
and-
bring the robots online?
Yeah.
Can we do it now? I'm done waiting. Let's
Science
have
Island. Can we go now?
I really need to go now. I wanna
Yeah,
go
can
there
I
so
go?
bad.
Can I run away and make this?
Look, this is what my people have been doing apparently for hundreds and hundreds of years. I had some. I was just looking at Anne Martha and Bouchier. Okay, she fled. No, her mother did. Her mother Mary fled France, went from England to the Americas to get away from the religious persecution of both of those countries. Then stuff gets a little too pilgrimy, puritanical in the east. They head to the Missouris. They head to the open lands, right? Give us a big science island in the middle of nowhere where nobody can see us, and we'll just have a proper educated civilization. And then, of course, the Civil War, and there's people. It's like, okay, California. All right, let's go to California. Keep running. Keep trying to get away from humans because they are absolutely insane. Yes, I want Science Island. I want it now.
So there was an
As a
experiment.
Son of the revolutionary.
Right, a son of the
Son of the American Revolution.
Right,
I
right.
deserve have another shot at this because apparently the first run failed. Look what's happened.
So he did an experiment with that he carried out, that other people have been carrying out online. It became viral video where he put water over cooked rice in 3 beakers and labeled one "thank you," one "you're an idiot," and then another one was unlabeled and ignored. And so for every day for 1 month, Emoto spoke whatever was on the bottle to the rice inside. And then after 30 days, his results were that the thank you bottle was giving a strong, pleasant aroma and fermenting well. And the "you're an idiot" rice turned mostly black. And the control rice also began to rot and turn disgusting. And so,
Okay.
yeah.
Sure.
Emoto says the ignored rice fared the worst because negligence and indifference are the absolute worst things we can do to water, rice, and ourselves. Not that that was your control and it just ruined your experiment. Anyway.
Also, if he never opened the control rice, that allowed the bacteria to grow very fast. Open it up, perhaps some bacteria escaped because you spoke into the rice and displaced some of the air. Right. And then also, maybe if you yelled, "You're an idiot," maybe some of your spit went into that container.
Karen.
No.
Oh,
yeah. Why don't you just label some rice Karen while you're at it? Karen.
Yeah. Bye, Replicant XI30 X3RO. It just hurts. It so hurts. Oh, Science Island. Oh, thanks for that link, Fada. Oh, good. They can get a lot of the science out of the ESA lander. That's great. Yay, the landing, even though it landed badly. That's good.
I feel like I might need to go soon.
Yeah, I should go too. I was just waiting. Hopefully, have we gone through? I should go to bed. I need to sleep. I must sleep. TWIS chat room. Why are you being weird? Okay, next Wednesday. We're not going to do a full show here. Spider! But we will join up and make sure we've got all our details covered, right?
Yes.
Go over a few things.
At 8 o'clock.
At 8 o'clock, yeah. And if people want to join us in the chat room, you can chat with us and we'll hang out and we'll talk Baltimore trip, but and talk plans for any additional plans that occur to us during the next week. I know, guest 24088, right? What is sleep? Sleep is something I do at night because I have a child that wakes up in the morning.
Facts. Contrary to popular belief, Science Island is actually a peninsula. Do you know that I used to think Rhode Island was an island? Because the way they would show the little line out, because it's so small that you can't really see it on a lot of maps. So they have a little, and the fact that it calls itself an island. Those two pieces weren't. Yeah.
So we're not doing like intro, outro next week on Wednesday or anything like that. We're just gonna chat on air for a bit. Or
Yeah.
Okay.
Yeah. Just say hi to people and remind everybody that, because if people show up and they forget that it's not the normal time, we'll be there. And we'll say, hey, this isn't the normal show, but this is what we're doing right now. And be sure to not forget Friday. Right?
Yes.
That sound good?
Absolutely.
Man, that hair, Blair. Is this the wig you were talking about? It's all
Which
choppy.
this
It's all
is
short and choppy. I love it.
it's just my hair now. I don't know what you're talking about.
Forever, ever. Yeah. All right, everybody. I can't think of anything else important that we're supposed to talk about right now, and consider but our show is coming up next week, and so I'm sure we'll consider something between now and then.
My gosh, you got a haircut.
Ed, is there a minion hangout tomorrow? Is Ed still here? Did he go to bed? Does Ed go to bed?
There probably will be.
Ed from Connecticut is not in the chat room. Yeah, tomorrow, scienceisland.org. Head over, see if there's a science minion, TWIS minion hangout tomorrow evening. And otherwise, everyone stay awake well, or sleep well, or do whatever you do well. And I hope that we will see you again next week. And especially we'll see you next week in Baltimore. Have a great week, everyone. Oh, wait, where's my Squirtle glasses again?